Hi students, so this video is about decoding a DNA sequence to its corresponding mRNA sequence to its corresponding amino acid sequence. Now my student asked me a question regarding this, so which we are going to discuss now, right? So let's move on to the question. A research group studied various aspects of a particular region in their gene of interest and its mutated versions. Now shown below is a part of the DNA sequence they were studying, right? So this is the DNA sequence that they have given us. So in the 5' 3' direction. So look at the direction, it is the 5' 3' direction. They have given a DNA sequence. Now we were asked few questions. So by answering each of these questions, we will be able to attain our understanding on how to decode a DNA to mRNA. Now what is the DNA sequence? The first question which was asked is what is the DNA sequence complementary to the above sequence? So the above sequence is this, right? It is from the 5' to 3' direction. So let's find out the complementary sequence. So we all know what adenine pairs with thymine and guanine pairs with cytosine, is it not? So let's start writing the complementary sequence. So this is from the 3' to 5' direction that I am writing. So adenine with thymine, T for A, and G pairs with C. Next T pairs with A, T pairs with A, G pairs with C, A pairs with T, yes, followed by G, T, and then A, G, T, and then A, G, G, then T, C, G, after which A, C and A. Yes. So I have written this complementary sequence in 3' to 5' direction, right? So look at the next question. Write the sequence of mRNA transcribed by the segment of DNA. So I have written both the 5' 3' direction, that the given DNA sequence and its complementary sequence, right? So now this 5' to 3' direction, this DNA sequence will be called as the coding sequence, is it not? It is the coding sequence or coding strand, and the other complementary sequence which is in the 3' to 5' direction is the template strand. Yes, so we all know that synthesis or transcription will take place in the 5' to 3' direction. So the template strand will always be in the 3' to 5' direction. So this will be taken as a template strand, and the complementary of this template strand will be our mRNA. Is it not? Will be our mRNA in the 5' to 3' direction. Also, you have to understand the coding strand. Yes, the coding strand. The mRNA which you have will be the exact sequence of the coding strand, but you have to replace the T with uracil because mRNA consists of uracil and not thymine, right? So let's understand or let's decode this. So how will you decode? You are going to synthesize the mRNA or transcribe, and the mRNA is synthesized in the 5' to 3' direction. So as you move from 5' to 3' direction, the sequence will be exactly that of the coding strand and complementary to that of the template strand. Yes, are you able to understand? So this the sequence of mRNA from 5' to 3' direction is what I am going to write now. So A, U, G, followed by U, U, G, A, C, A, U, C, A, U, C, C, A, G, C, UGC. Sorry, UGU. Yes, so this is the mRNA which we will synthesize. This mRNA, if you can look at this, it is exactly that of the coding strand when when the thymine is replaced by uracil. Yes, and it is complementary to that of the template strand. Yes, can you see? It is complementary to that of the template strand, but whereas it is exactly that of the coding strand with U replacing the T of the DNA, right? So this is the mRNA sequence. So next, write the amino acid sequence coded by the mRNA sequence using the genetic code table. So now this is the mRNA sequence that we had arrived at. So if you can decode the amino acid sequence from this genetic code table, what will you get? AUG is methionine. Yes. UUG is leucine. ACA is threonine. UCA is serine. UCC is also serine. AGC is serine. And then UGU, yes, UGU is cysteine. So this is the amino acid sequence with N-terminal and C-terminal. Yes. So next we'll move on to the next question. A mutant of this gene contains A instead of C in the 11th position in the given DNA sequence. So we had been given a DNA sequence, is it not, in the 5' 3' direction. So in that the 11th position had C, but instead of C, A is been replaced. Yes. So if it is been replaced or if it is mutated, what will happen to the mRNA sequence? What will happen to the amino acid sequence? Now since this is in the 5' 3' direction, this will be the coding strand, is it not? So how will you write the mRNA sequence? You will write the mRNA sequence by replacing T with U, right? So I'll write this AUG, UUG, ACA, and what will you get here? UAA, UCC, followed by AGC and UGU, right? So this is the mRNA sequence. Now what will be the amino acid sequence? All the amino acid sequence will be the same. What has happened in the mutated area? It is UAA. Yes, what is UAA? It is a stop codon, is it not? What is UAA? UAA, UAG and UGA are stop codons, is it not? So it is UAA in the 4th codon. Okay, so that has become a stop codon here. So what has happened here? What kind of mutation has happened here? Since it becomes a stop codon, the protein synthesis is truncated at this level, is stopped at this level. So a truncated protein is being synthesized, which leads on to a nonsense mutation. Whenever an amino acid is converted to a stop codon on mutation, that becomes a nonsense mutation. Yes? So hope you have understood. So this is how you decode a DNA sequence to its corresponding mRNA sequence and then to its corresponding amino acid sequence. Thank you all.